Hello everyone, and welcome to this Integrated DNA Technologies webinar on increased efficiency of genome editing with the Alt-R CRISPR-Cas9 System, design and use. My name is Dr. Hans Packer, and I'll be serving as the moderator for today's presentation. And the presentation today will be given by Dr. Garrett Rettig. Garrett is a research scientist here in the molecular genetics group at IDT. Garrett has his PhD in pharmacy medicinal chemistry from the University of Iowa, where he studied synthetic peptide delivery systems for plasmid DNA and siRNA, both in vitro and in vivo. At IDT, Dr. Rettig has been extensively involved in functional genomics research, including high-throughput screening of siRNAs in vitro, and more recently, CRISPR-Cas9 genome editing methods. Garrett's presentation today should last about 35 minutes, and following the presentation, Garrett will answer questions from the audience. As attendees, you've been muted, but we encourage you to ask your questions or make comments at any time during or after the presentation. And you can do that by typing them into the questions box, and you'll find that located in the GoToWebinar control panel. If you look down about halfway in the control panel, you'll see questions, and you can click the little up arrow or plus sign depending on whether you're in a Mac or a PC. That'll make that window larger, and you can type your questions into that. Also, in case you need to leave early today or you want to revisit this webinar at a later date, we are recording the presentation, and we will make the link to the recording available within a few days of the presentation. And we'll send you that in an email, or you can just visit our YouTube channel, www.youtube.com/IDTDNABio. We also have our recent webinar up there on new RNA tools for optimized CRISPR-Cas9 genome editing, and that's the precursor webinar to this, where we kind of go through the research that led to the development of the system. The slides for this presentation are actually already available on our SlideShare site, so you can check those out after the presentation, which is www.slideshare.net/IDTDNA. If you didn't get any of those links, don't worry about it. We will be sending you an email with links to all the relevant materials. And with all of that housekeeping stuff out of the way, I will pass this over to Garrett and let him start his presentation. Garrett?
All right, thank you, Hans, for the introduction. As he was mentioning, this is certainly a follow-up to Ashley's recent webinar. Ashley gave more of an in-depth discussion of all the data and all the research really that led up to launching of the Alt-R CRISPR products. In this particular presentation, it'll be the way forward focused more on exactly how to use the Alt-R CRISPR product, particularly if you're new to the idea of using CRISPR-Cas9 for genome editing. I do think this webinar, in tandem with Ashley's, provides a good basis for those just entering the field, and as well provides meaningful insights, hopefully, to those that are more expert in the field and have experience with the CRISPR-Cas9 system. With that, let's begin then the discussion of the Alt-R CRISPR system. So this is a nice slide, I think, to highlight, first of all, at its core, the simplicity of the CRISPR-Cas9 system for gene editing, that is, requiring the Cas9 protein as well as the CRISPR guide RNA. But then when you start to look into it a little bit more and think about the possibilities for experimentation, you start to see that there are many options for delivering the Cas9 protein, whether it's from an expression vector, be plasmid-based or lentiviral, via the messenger RNA, developing a stable line, which constitutively expresses Cas9, or delivering the Cas9 protein itself in complex with the RNA as a ribonuclear protein complex. By the same token, there are many options for delivering the guide RNA, the CRISPR guide RNA that directs the Cas9 endonuclease to the specific site for editing in the genome. One would be, once again, an expression vector that expresses the single guide RNA. This could be done from a plasmid or a G-block gene fragment from IDT. In addition, some other common uses are using in vitro transcribed single guide RNAs, once again, as a chimeric molecule, combining the CRISPR RNA and the tracer RNA into one single guide RNA. And then what we focused on here in leveraging our institutional knowledge as well as our expertise in synthetic oligonucleotide research was to develop a two-part system. which is reminiscent of the native system used in strep pyogenes, which includes a synthetic oligo for the CRISPR RNA, which is going to be a custom, containing a component of a custom sequence that will direct the endonuclease to a specific location in genomic DNA or double-stranded DNA, as well as a universal tracer complex, which can be paired with any of the CRISPR RNAs. And just to add to the complexity as you're thinking about experiments, there are all the options for delivering Cas9 as well as the guide RNAs, and we haven't even considered yet routes of delivery, whether it be viral or lipofection, nucleofection, microinjection, etc. So bear in mind, we're focusing here mostly on, first off anyway, the delivery of the synthetic oligonucleotides and development of that system. Shown in the panel at left, just highlighting the synthetic oligonucleotide that comprises the crRNA or CRISPR RNA, as well as that comprises the tracer RNA. As I mentioned, the crRNA is a custom oligonucleotide. The end user would come to us with the 20 nucleotides or 19 nucleotides of specificity for genomic DNA, and then there would be an appended additional 16 nucleotides and a 3' end. As we've done the work to trim this down to a 36 nucleotide oligo, it represents one that we can reliably and robustly synthesize as a custom oligonucleotide. The tracer RNA, it's also meaningful that we've trimmed it down to a 67 nucleotide that actually contains rather heavy base modifications to afford various nuclease protections. But this is, again, a universal sequence that we can synthesize in large batches, which aids in the manufacturing process. So these two components that come together represent our Alt-R CRISPR RNAs. The panel at right shows a comparison in the far right, in the green bar, the results from gene editing at one particular site with our Alt-R CRISPR RNA tracer RNA complex. This was compared directly to the native two-part system from strep pyogenes, which is shown in the purple bar, as well as compared directly to the single guide RNA, a G-block gene fragment, or a single guide RNA driven from a plasmid expression vector. And this comparison was made across dozens of sites, not just one, and showed that the Alt-R CRISPR RNA tracer RNA complex was superior throughout, and a subset of these different RNA guide sources were not only compared across dozens of sites, but hundreds of sites, which gave us the confidence that we're not looking at something that's site-specific here, that we are seeing something that is universal to the length-optimized Alt-R CRISPR RNA tracer RNA complex. Slide 4 gives a little bit of insight of what was tested then in terms of looking at trimming down the tracer RNA from 89 nucleotides down to 63 nucleotides, as well as comparing at each length in length optimization studies. And again, this is for one specific site, and these experiments were done in our HEC293 cell line that stably expresses Cas9. We compared 42 nucleotide CRISPR RNA down to 34 nucleotide CRISPR RNA, and we observed that the 36 nucleotide CRISPR RNA paired with the 67 nucleotide tracer RNA gave us the most potent results in gene editing. So in addition to the two Alt-R products that we've talked about so far, the CRISPR RNA and the tracer RNA, just by way of introduction, I also want to mention that additional products in the Alt-R line are HBRT positive control kits, which include a 2 nanomole CRISPR RNA for human, rat, and mouse. That kit would also include a negative control, 2 nanomoles of a negative control CRISPR RNA, as well as 5 nanomoles of a tracer RNA. These components can also be purchased individually. The HBRT positives for human, rat, or mouse is the 2 nanomole CRISPR RNA. And we also have 3 sequence options for scrambled negative controls that have all been bioinformatically screened against human, rat, and mouse genome to have, so as to avoid off-target effects in any of those mammalian genomes. We also include, for the positive controls, the species-specific PCR primer mix for amplification of the target region within HPRT for the downstream detection of editing by a T7 assay. And then finally, we do offer the Alt-R Pyogenes Cas9 expression plasmid, which is a stripped-down minimal plasmid, size is about 7 kb for expression of Cas9. Then some general guidelines as the end user may be entering into thinking of ways to incorporate or reasons to incorporate gene editing experiments into their own research. What we want to mention here, really before beginning the experiments and prior to ordering, certain considerations that need to be made in terms of the CRISPR RNA. What is the genomic target going to be? Where within the genome are you going to target? Is the intention of the genomic editing for protein disruption? If so, which exon within the gene is going to be targeted? Or rather, is the intention with the double-stranded break in the genome that would be mediated by Cas9 and the Alt-R CRISPR RNAs, would it be to target a non-coding genomic region for homology-directed repair to paste in a DNA template? As well, what is the availability of PAM sites within your particular region of interest? Of course, the Cas9 recognition is dependent upon the presence of an NGG that is 3' to the specific 20 nucleotide double-stranded DNA segment that's targeted by Cas9. And then other considerations when planning the Alt-R CRISPR-Cas9 experiments would be what cell line is going to be used and what other associated reagents are required. It should be said that our protocols are based upon our own in-house research, and we do make specific recommendations for delivery of for instance, the Cas9 expression vector as well as the Alt-R CRISPR RNAs. But that's not to say that these recommendations are not for products that are IDT products. They have just been for ones that work best in our hands, so there may in fact be room for optimization in your own hands in your own specific experiments. And what other reagents might be necessary as you go into this for downstream detection of edits, whether it's specifically looking for editing by sequencing by the T7 assay, or is it a phenotype readout that's going to be looked at. Highlighted in the panel in terms of design, and the panel on the right side of the screen shows what we've done to empirically determine in a particular human exon, and I think this is the case of STAT3, one of the STAT3 exons that we looked at. One of the reasons that we haven't focused too much on design rules for a positive CRISPR RNA is that we see with relative consistency across 6 different exons that we've looked at in 4 genes, that we see good editing efficiency from most of the CRISPR RNAs, the crRNAs. That being said, this is why we then recommend typically testing 2 to 4 crRNAs in the genomic region of interest in order to ensure that there is at least one with very good editing performance. We feel pretty confident about that. The manuscript listed below from 2014 goes on to actually make some additional recommendations that I would just highlight here. These are some simple design rules that came out of this manuscript for protein disruption based upon their validations. They showed increased functional editing when their guide RNAs were targeting coding regions that were at or adjacent to exon-intron junctions. They came away with recommendations to avoid the 5' and 3' untranslated regions of the gene. And also, I think it makes sense that as you move further towards the C-terminus, that it reduces the hit rate in terms of generating edits or mutations that actually frame shift and produce a non-functional protein, if that's the end goal. And finally, one thing that we've actually implemented in our work, as well as to look at an exon that is at the more 5' end of the gene that is about 1 kb or greater, so that we can, in our case, we were wanting to design an assay that was about the size that we could look at, randomly look at all the PAM sites within there, and targeting, again, at the 5' end so that we can specifically make the edit more towards the N-terminus of the protein and frame shift and cause a frame shift in order to knock out the protein. Regarding design to avoid off-target effects, we are currently suggesting to use the free tool found at crispr.mit.edu. With this particular tool, the input is 250 bases of the double-stranded DNA target. This can be screened against 16 unique reference genomes that are present that you can select at this particular website. And it also has a batch entry option if your region of interest in the region that you want to search is greater than 250 bases. You can paste several in and do parallel searches in order to segment out those 250 bases into a larger region of interest. The scoring readout is based off of work from the 2013 Nature Biotech paper from Su et al., which considers the mismatch, the position of the mismatch, or the location of the mismatch within the guide RNA, as well as the distance between those mismatches, and all, again, then coming together to bioinformatically at least try to eliminate the most off-target effects as possible. So this screenshot it captures a nice image of what is shown from entering a 250 base double-stranded DNA fragment into the tool. It highlights every PAM site and the protospacer element that is directly adjacent to the PAM, and it also orients them in terms of whether it's on the antisense strand or the sense strand. So you see the directionality of the protospacers relative to their PAM sites as well. And then the results really are represented here. And the color coding is nice. It represents in green minimal predicted off-target effects for these particular protospacers within your region of interest. The yellow indicates guides that should only be used when there are other better alternatives not available. And red represents guides that should be avoided. And what you can learn additionally by hovering over any one of the guides, in this particular case, this represents the readout for guide number 2 listed at left. We see the specific off-target sequence that may be targeted by this particular 20 nucleotide protospacer. It tells you the score for that specific off-target site. It also tells you exactly, well, how many mismatches there are, where the mismatches occur within the guide. Also, it will annotate which gene this potential off-target site is present in, if it is, and it also specifically annotates the locus, chromosome number, and position. So really a nice tool that that we use in-house and recommend others to use as well. And then, as I was mentioning earlier, the relative efficiency, good efficiency that we see when randomly testing the Alt-R CRISPR CRNAs. And again, these experiments were done in our HEC293 cell line that are stably expressing Cas9. What we observe is about 85% of the CRISPR RNA sites that give greater than 20% editing as measured by the T7 assay. And again, I think it bears mentioning that these are not selected, these are not pre-selected at all for by any algorithm that would have predicted potent sites. These are just, we simply designed PCR assays to be used in the T7 endonuclease mismatch detection assay, and really represent then random distribution of all sites within those PCR amplicons. And what we observe then is about 85% of the sites that give 20% editing by T7 cleavage, which we know to represent 40 to 50% editing when we actually look at the sequencing at the level of Sanger sequencing. And once again, this is why we recommend selecting 2 to 4 CRRNAs within your region of interest to test to find one that would give good function in genome editing. Then ordering CRRNAs, ordering the Alt-R CRRNAs. This graphic, I think, does a nice job of highlighting down below in red the common errors that can occur. Specifically when we want to design a CRRNA, take the sequence of genomic DNA and excluding the PAM, the NGG that is highlighted in the orange color at the top, it is the 20 bases that are directly 5' to the PAM site. So the guide RNA is identical to the DNA sequence that is directly 5' of the PAM. It does not include the NGG sequence, as shown below, crossed out. It is not the reverse complement of the target. It's not the reverse of the target, nor is it the complement of the target. The guide RNA then ends up being complementary to the strand of DNA that is opposite the PAM site. So what we allow for then is the input in the ordering page of the 20 or even 19 nucleotides that are directly adjacent to the PAM sequence. And as you might be thinking, then the 20 custom nucleotides then in our configurator ordering tool is automatically appended with the additional 16 bases on the 3' end of the Alt-R CRNA, which will yield a 35 or 36 nucleotide CRRNA as we discussed before. One thing to consider in further consideration when selecting a CRRNA is where it is in relative location to the primers of the PCR assay that's been designed, if the end readout is the T7 assay, and typically ours have been, so we kind of view everything through that prism. But what can happen is that you can get some editing events, and if the editing event of the CRRNA directed too close to the primer binding site could influence the binding and inhibit really the binding if the deletion or insertion is too large of the primer. So that needs to be considered. In addition, we've looked at whether or not, and others have as well, the GC content as it relates to the impact on function and the particular orientation of the PAM, whether it's on the sense or antisense strand. We have not seen either of these factors to impact function. What is relevant, though, to mention is that one, whether you're on the sense or the antisense strand, will influence the orientation, of course, of the protospacer relative to the PAM site. And the GC content can influence the frequency of PAM sites within a particular region of interest. So if your region of interest has a high GC content, we haven't seen that to bear out to give more or less potent CRRNAs. It just means that there are more total CRRNAs between that high GC, in that high GC region, as there would be in a low GC content region, of course, because the PAM site is dependent on the presence of an NGG. The scale for the CRRNAs that are offered are 2 and 10 nanomole deliverables, and there's also a batch entry option in which at the time of order you can order 2 nanomoles of CRRNAs on a plate as long as you make a minimum order of 24 CRRNAs. The ordering site then can be found at idtdna.com/CRISPR. The website here for our Alt-R CRISPR CRNAs should look and feel very similar, and does, I think, to our typical ordering interface that you would see in general for IDT oligos. And what you'll notice below is that you can directly grab in the sequence box, you can directly grab the DNA sequence that you wish to target that is directly adjacent to the PAM site, paste that in, and click the Convert to RNA button, and it will directly convert all the DNA bases to RNA. And then, as I mentioned, there's the 20 nucleotides that you enter. This additional 16 at the 3' end of the CRRNA will be automatically appended here in the configurator tool. plate option allows you to orient, again, at a minimum of 24 and up to 96 crRNAs on one plate at 2 nanomole, and does afford a discount over ordering the same Alt-R CRNAs in the tube, and allows you to manipulate them across the plate and order them as you wish. Once again, the Alt-R CRISPR tracer RNA component is the universal sequence. It's a 67 nucleotide, rather heavily modified with a proprietary chemical modification pattern. Those mods do confer increased nuclease resistance. We offer the tracer RNA at 5 nanomole, 20 nanomole, and 100 nanomole amounts. One thing to be considered at the time of order is what is the application of the CRISPR RNA tracer RNA complex? Is it going to be in tissue culture transfections in a 96-well plate? If that's the case, a 5 nanomole deliverable will allow for about 1,000 96-well plates to be transfected, and scaled from there. That's with liposuction. If the same experiments are looking to do with nucleofection, that can require a couple orders of magnitude more reagent in terms of the CRISPR RNA tracer RNA complex. And so you'll want to scale your orders appropriate to the application of your editing experiments. Ordering controls. Like we mentioned earlier, these are available both as kits. The kits would include a species-specific HPRT CRISPR RNA, as well as a negative control CRISPR RNA, crRNA, as well as 5 nanomoles of a tracer RNA, and a primer mix for the species-specific primer mix for the HPRT positive control detection. Those are, once again, available as kits or as individual components. The panel at right shows just a representative editing that we see by T7 Endonuclease I cleavage assay for human HPRT in HEC293 cells, mouse HPRT in HEPA16 cells, and rat HPRT in RG2 cells, where we see the full-length product, PCR product, in each case is about 700 nucleotides, and we form heteroduplexes to a degree that they're recognized by the T7 Endonuclease and cleaved, and the resulting products are shown there that are quantified and here show validated positive controls for these 3 species. And then additionally, the final component of the Alt-R product line is the strep pyogenes Cas9 expression plasmid. As was mentioned earlier, this is a trimmed down vector, so we don't have any GFP or orange fluorescent protein that would be suitable for sorting the cells that are transfected. We've also stripped out any G418 or any other selection markers. But what we've seen by doing this is that we've actually been able to show improved editing efficiency over other commercially available plasmids that are larger, more on the order of 10 kb. The deliverable here is 1 microgram of dry plasmid that, once again, depending on your application, but most likely would need to be propagated in competent E. coli for additional downstream experiments. And speaking of those experiments, this is, in the panel on the right, represents the cover page for, I think, a really nice user guide that we've developed between our research group and our scientific communication group for executing CRISPR-Cas9 genome editing experiments. The first step, as I mentioned, is to take the 1 microgram if you're going the route of using an expression vector, as in the Alt-R pyogenes Cas9 expression plasmid, growing it up in competent E. coli, and using standard laboratory techniques to grow up and purify plasmid. What we recommend for then transfecting the plasmid into mammalian cells is to use a 100 millimeter dish and use the Transit X2 transfection reagent from Mirus. We recommend using 15 micrograms in the 100 millimeter dish, 15 micrograms of plasmid, and 45 microliters of Mirus X2. If you need more or less, if your cell needs are greater or less than really what would be yielded from a 100 millimeter dish, then the Mirus protocol as scalability that would discuss amounts of plasmid as well as amounts of the X2 to be transfected to optimize the conditions for something that was maybe done in a larger flask or done in 6-well plates or 24-well plates, whatever the case may be. We do the plasmid transfections as a forward transfection. That is, the cells are plated 24 hours prior to transfection, so that, or at least overnight, so they're allowed to become adherent to the plate and reach about 70 to 80% confluency, in this case, once again, in the 100 millimeter dish. And as part of this, we make up the complexes, the Transit X2 plasmid complexes in 1.5 milliliters of Optimem, transfect the adherent cells, and incubate for 24 hours. One thing that's mentioned here is that point number 7, the removal of media after 6 hours and replacement of the transfection media with complete media. In most cases, in most of the mammalian cell lines that we've tested, we found this not to be necessary, but we include it. And then once again, this is a 24-hour transfection to allow for expression of Cas9 protein after 24 hours in these cells. So on the panel at left, first we talk about resuspension of the crRNA and tracer RNA that are received from IDT. These resuspensions, we call for them to be done in TE buffer as a 1x diluted to 100 micromolar final concentration. And we've got a resuspension calculator on our website, idtdna.com/scitools. These we typically store with great reliability and stability at -20 degrees Celsius. What we recommend then for a working dilution is to do a 1-to-1 mole-to-mole ratio dilution down to 3 micromole of the Alt-R CRNA tracer RNA complex. This makes it convenient for doing transfections in 96-well plates that are on the order of 10 nanomolar. Specifically, we're talking about doing transfections at 30 nanomolar. Points 3 and 4 actually, recommend using a 95-degree heat step in order to form the complex or form the duplex domain between the crRNA and the tracer RNA. We actually just looked at some data this morning where this may not be necessary. So I just want to point out that the heat step may not be necessary. In addition, we call for using duplex buffer. It may also be true, and we've shown this in at least in one experiment, that we can do the duplexing in TE buffer, which really speaks to the point that these are an evolving set of experimental protocols that, based on your feedback, based on customer feedback, based on things that we continue to try to improve in our lab, that these are evolving a little bit dynamic. But these are, in fact, 2 bits of data that we just saw this morning from some experiments that don't necessarily call for the heat step or for using duplex buffer. But regardless, the crRNA and tracer RNA complexes are then lipoaffected into the cells that are now expressing Cas9. We recommend, as I mentioned, a 30 nanomolar transfection using lipoaffectamine RNAi max. And typically, we find this to be optimal at 0.75 microliters of RNAi max per well in a 96-well plate. And as I mentioned earlier, and the same is true for the Transit X2, there may be some tweaks, modifications to the protocol that that would be more optimal, that could be made to optimize your specific experiments in your specific cell line in our hands, in HEC293 cells, HeLa cells, HEPA-16 cells, rat RG2 cells. These are the conditions that we have shown to be optimized. And one important thing I think to mention here as well, we've talked a lot about the positive control and negative control offerings of crRNAs, 2 nanomole deliverables as IDT products. In addition, it is just good experimental practice to include maybe the positive controls and tease out some of these variables and tweaks and modifications that can be made to the protocol, whether it's a dose response or whether it's of the crRNA itself, ranging around 30 nanomolar, or whether it's a dose response with the liquefaction reagent ranging around plus and minus 0.75 microliters. Also, the negative control is important to consider, and not just the negative control crRNA from the kit. Likewise, other negative controls such as a cells-only treatment, a reagent-only treatment, and even transfections done with the Alt-R CRNA tracer RNA complex into non-expressing Cas9 cells. That is, those were not transfected by the pyogenes plasmid. That will allow the end user really to discover any sensitivity that the cells might have to any of these treatments, and whether or not that sensitivity, if the appropriate controls are included, whether or not that sensitivity is specific to an editing event or simply the delivery of the reagents themselves. So we really recommend, and any further follow-up with our scientific applications group would really require assistance to be directed based on what you see with your positive and negative controls. So we recommend those good scientific practices. And then one thing to highlight in the last point on this slide from the protocol, point 6, is that this is a 48-hour transfection. Consistently, we've seen that reverse transfection over 48 hours has been optimal, and it doesn't really improve in terms of the gene editing that's observed after 48 hours. But up to 48 hours, we do see increased gene editing efficiency, which is why we typically do these transfections for 48 hours. After 48 hours, then we want to be able to discern how much editing we've observed. And just as an overview, as I've mentioned throughout, our assay of choice, especially as it relates to looking at almost 600 sites, looking at 600 crRNAs across 6 different amplicons, we've chosen to look at editing with a T7 Endonuclease I assay. The steps for this, of course, are to, as we mentioned, define a PCR amplicon, and we get these amplicons that contain either a wild-type sequence, if there was no double-stranded break that occurred in that particular cell or that particular locus, or if non-homologous end joining corrected the sequence to wild-type. We could have the presence of some wild-type there in our PCR amplification. Other possibilities following the double-stranded break when non-homologous end joining pathways take over are for insertions as well as deletions. If we form heteroduplexes by heating to 95 degrees and then doing a slow cool, we will get formation of heteroduplexes. We'll also get wild-type double-stranded DNA that's formed, and maybe some single nucleotide insertions or deletions that wouldn't necessarily be detected by the T7 E1 assay. But largely, what we see is a pairing of a sizable insertion. Maybe it's 5, maybe it's 20 bases, paired with a wild-type strand. Likewise, a wild-type could pair with a deletion event, so forming a bubble then on opposite sides of the wild-type strand, and the deletion could pair with the insertion. All of these, shown in the figure, would be recognized by the T7 E1 mismatch endonuclease and would be cleaved. And our final readout then is an electropherogram in which we see a full-length PCR product. Those would be the undigested products at the top, and then we would see the cleaved fragments that would have sizes specific to the site of crRNA double-stranded break. And in order to execute a T7E1 assay, We've already talked about including the primer mix for the HPRT positive controls for human, mouse, or rat. In addition, we would direct the end user to design a PCR assay using our Primer Quest tool by entering your region of interest and then using this fairly straightforward tool to design primers that would be then used in the T7 assay. Our recommendation here is that the end user generates somewhere between a 600 to 1,000 base pair PCR amplicon around the region of interest, with approximately 100 base pairs flanking the outermost CRISPR cut sites within that amplicon. As mentioned before, this would eliminate any complications of edits as it would impact the binding of the primers for amplification. In addition, it allows for fragments, detectable fragments of 100 base pairs or larger to be resolved on a gel. And speaking of gel and detection methods for the resultant products of the T7 assay, this is really an overall workflow of what we would typically do from transfection through genomic DNA extraction, for which we use the quick extract buffer from Epicenter. We would then heat at 65 degrees and then followed by heat inactivation of proteases and RNases at 95 degrees. And then we would amplify our specific genomic DNA region of interest. We recommend using the kappa hi-fi polymerase. And then directly go into forming our heteroduplexes from the PCR amplicons of our cell population that has been edited. So once again, we would anticipate having some wild type as well as some insertion and deletion and want to get those heteroduplexes formed. So that's a heat to 95 degrees and a slow cool to allow for heteroduplex formation. We would then introduce T7 at 37 degrees, 2 units of that for 1 hour. And then we would take these resultant fragments and load them onto our tool of choice, really, and one of our favorite tools in the lab, the Fragment Analyzer from Advanced Analytical. This is a 96-channel capillary electrophoresis instrument that can complete a run in about an hour. It can resolve fragments from 10 to 40,000 base pairs, and not only can it handle 196-well plate at a time from which it will sip reagent and run through capillary electrophoresis, but it also we can load up to 3 plates at a time, which really lends well to high throughput usage. We also only have to generate about 1/10 of the PCR amplicon to be able to reliably and robustly visualize and quantify the digest results on the electropharogram. What you can see in the center of the panel here is the full-length PCR product and the area under the curve highlighted in red, and then the 2 specific cleavage products and their areas under the curve integrated and highlighted in orange. So we've really grown to appreciate this instrument and have moved away from running agarose gels and do our detection and quantification of our editing events by T7 looking at them on the Fragment Analyzer. Additional resources for your CRISPR-Cas9 experiments can be found once again at idtdna.com/CRISPR. There's a support tab that contains the user guide, which we highlighted, as well as Ashley's previous webinar on a more in-depth data presentation that led to the development of the Alt-R CRISPR-Cas9 system. Additionally, there are short tutorial videos for ordering on the support tab. The performance tab highlights some of those key pieces of data that Ashley would have talked about in her webinar as well. I mentioned this group earlier, but the application support group is available for web chat, email, or phone call to help you troubleshoot or help you design experiments. And thank you for listening. Thank you also to the scientific communication group for putting together the webinar. As Hans has mentioned, the webinar will be available. The slides are already available on the website, so you can access those immediately. And with that, I would take any questions you might have.
Okay, thanks for the great presentation, Garrett. We do have a bunch of questions already, but I just want to let people know if you haven't asked your question already and you'd like to do so, you can type that into the questions box, which is in the GoToWebinar control panel, and you'll find that about halfway down there's a little plus sign or an up arrow in the bar for the questions panel. Just click that, make it larger, type your question in, and I will ask Garrett as many of them as I can. So let me get started here. So I'm not sure if you can answer this first question, Garrett, but do we have any information about using the Alt-R CRISPR system in human iPS cells?
We have several beta test sites that are looking at anywhere from zebrafish to mouse embryos for implantation. I do not specifically remember if we have a beta test site with human iPS cells. I'm guessing that this end user would want to look at probably electroporation. In those cases, we are actually actively trying to look at a collaboration in that regard, but existing now, I don't think we've actually tested the Alt-R CRISPR-RNAs in those cells.
They have tried a larger plasmid, and it's from the Shang lab, it sounds like, than what we offer, and I'm just wondering if there's anything to suggest that maybe the smaller plasmid that we use would work better, but you mentioned electroporation, that might be a better.
Yeah, smaller plasmid perhaps could work better, as well as electroporation, and then perhaps that's with a ribonuclear protein complex to get optimal results there.
Okay. So with the difference in size of the Alt-R CRNA and tracer RNA complex, do we see any change in off-target effects? Do we have any data on that?
That's a great question. We are actively efforting to determine that an answer to that exact question right now. We do not have any data.
Yeah, the methods for determining off-target effects are complicated. It's not an easy question to answer, I know. So also regarding designing the CRRNA sequence, is there any plan for IDT to offer a design tool for the Alt-R CRISPR CRRNA?
There is, in fact. We've had our bioinformatics group has done a great job in terms of developing algorithms based upon data for effective and non-effective for looking at data sets of siRNAs and dicer substrate siRNAs specifically that we've looked at and been able to develop algorithms that can reliably select the most potent siRNAs. We're using some of the same parameters to train an algorithm with our large data set that would be offered as well for designing and selecting CRISPR RNAs. Yes.
Do we have any experience to say whether the Alt-R CRISPR system will work with Cas9 mRNA or Cas9 protein delivery?
Yes, we definitely have experience to suggest that it does work and does work well with protein delivery. And just to circle back around to the notion of the ribonuclear protein complex, this is something that, in addition to the algorithm, that we are actively pursuing and anticipating launching a product soon to offer the Cas9 protein as part of the Alt-R product line as well. Sorry, the other component of that, with the mRNA. Yes, with the mRNA, we have some beta test sites that have looked at that. We have also looked at it internally, but not to a degree that we can make any sweeping statements about it. But our beta test sites have had success using the Alt-R RNAs with Cas9 mRNA.
Okay. So we have shown some data, a little bit in this webinar, but also in previous webinars, about efficiency of the Alt-R CRISPR system. Is there anything about improved specificity with the Alt-R CRISPR system?
Yeah, well, and the improved specificity, right, goes to the question of does it reduce off-target effects a little bit? And I mean, yes, we show more potency on target, but we have not investigated those results at off-target sites yet.
And I'm actually going to ask a related thing. I'm going to jump ahead here just a little bit. When you were talking about what we allow people to enter into the ordering tool, is there any How does a person know when to choose a 19 base pair target or a 20 base pair target for their CRRNA? Is there a reason to choose 19?
Mm-hmm. Yeah, that question came up because there was a publication from the Keith Young lab that suggests going down to even a 17 nucleotide guide RNA actually can maintain potency on target and dramatically reduce off target because you trim the bases that are most 5' PAM distal sites. What we've found empirically in-house is that trimming to 19, we haven't, again, don't have any off-target data, but trimming to 19, those CRRNAs really held up well when compared to the full 20 nucleotide, 20 plus 16, 36 nucleotide CRRNAs compared to the 35. But when we went to, so when I say they held up well in terms of on-target potency. When we trimmed further to 18 nucleotides, the potency on target dropped about 50%, and when we trimmed to 17 for the set that we were looking at, which was either 12 or 24, I can't recall, but the potency pretty much dropped down to 0 on target. So that's why we made the decision to allow the input of 20, which would be the canonical choice, and then trimming to 19 if someone wanted to do that in order to perhaps reduce off-target effects based on the suggestion of that particular manuscript. We would allow those because we do still see pretty good potency with the 19 nucleotide plus the 16 for complementarity to the tracer.
Okay. Have we done any testing with the Alt-R CRISPR system in vitro, just using the components in a tube or just in cells?
Yes, we've used them in a tube. We see quite reliable cleavage, full cleavage done in a tube, and in fact we first started testing different length modifications and even chemical modifications of the crRNA and the tracer RNA in a test tube, so to speak. But then we saw incongruous results when we transferred these same length and chemical modified RNAs over to a cell-based system, and that was basically when we determined that we would go forward with all testing in cells in order to, get rid of any of these idiosyncrasies between the two different experimental platforms that were kind of confounding our results. What really we determined really mattered was their function inside of a cell, and so that's why we turned to doing the high throughput screening.
So the in vitro system, the results were pretty good for a variety of different constructs, but it was really
they're
in
quite
cells
good. Yep.
where the Alt-R system shows its benefits.
That's correct.
Okay. This is a very specific one. I don't know if you'll have something for this, but we'll try. Can the Alt-R CRISPR system target highly repetitive sequences such as telomeric DNA or other G-rich repetitive sequences?
Can they target repetitive sequences? Well, the answer would be yes, but targeting a repetitive element would have the inherent risk of off-target effect. We haven't specifically tested that, would be the short answer.
Okay, we've covered this. Let's see here. Do we know anything specifically about whether or not Cas9 is, whether it's a multi-turnover enzyme or a non-turnover enzyme? I had to ask for a little clarification on the usage of the term here, whether or not Cas9 is basically able to cleave one site, and then if there's another site that, it also recognizes, can it cleave more than one site within the cell?
Right. Early on in the discussion of Cas9 and the CRISPR system, it was suggested, and I can't recall which group, what manuscript specifically noted this, but it was a single turnover event. And we haven't done any specific experiments to follow up on that.
Okay, so we don't have any data. All right. Specific protocol question here for you. Is the nuclease-free duplex buffer the same for the CRISPR, the CRRNA, tracer RNA complex as it is for siRNAs?
That's correct. That's the same 30 millimolar heat piece buffer and I think 100 millimolar potassium acetate pH 7.5 that we offer and use consistently with the duplexing of the dicer substrates.
Okay. There's two related questions here. The first one was, for expressing Cas9 in your cells, how do you recommend to measure your Cas9 expression in transfected cells? And then another question is there a level of Cas9 expression that is required for optimal CRISPR activity?
Okay, those are two good questions. We have done this in, well, a couple different rounds of experiments. We've not done it as much. In our particular plasmid system that we discuss here as part of the Alt-R product line, we've just looked at functional results of editing. But in our stable cell line, our HEC293 cell line that is stably and constitutively expressing Cas9, we have designed qPCR assays to isolate total RNA from these cells and look at the relative expression levels of Cas9. In addition, we've done droplet digital qPCR to look at total copy numbers and have determined with the ratios to other housekeeping genes that we have one single integrant in our Cas9 stable cell. So short answer, and also based on Western blot analysis that we've done comparing this type of transient expression to what we see in our stable line, we've seen a very, very low level of Cas9 to actually be detected and expressed in the stable line, and still that amount is enough to generate editing that is just as good as what we have seen in our transient transfections, which really produce a lot of Cas9 in terms of what is actually shown by protein. So we do think it is a very small amount that is actually necessary to do the editing within a cell.
Okay. This next question is, could you comment on the feasibility of incorporating the Alt-R CRISPR design strategy in a pooled screen, and then it says 500 to 1,000 CRNAs per gene, for example, or 500 to 1,000 genes?
So pooling 500 to 1,000 CRISPR RNAs in a single experiment and then doing the transfection? I guess I'm not exactly sure.
Yeah, yeah, I guess we'll have to follow up with them. Maybe they can clarify their question a little
Or
bit.
perhaps they're discussing a screening library. I'm not sure what exactly the question is.
So is there any plan to develop this system for other organisms?
We have not specifically in-house capability really to develop for other organisms. I think I was mentioning, maybe it was in an earlier webinar, that we have beta test sites that are looking at zebrafish, certainly. And it is the same, it's the same components from this paginy system that are translated into, in this case, mammalian cells or zebrafish or plants or whatever. So we would anticipate that the Alt-R CRISPR products would work cross species. And have to this point observed exactly that.
We're getting close on time, but I'll try and squeeze in one or two more questions here. So, our lab has already invested some time into designing G-Blocks gene fragments for single guide RNAs, and they want to know if it would be good for them to switch to the Alt-R CRISPR system before they make too much progress.
No, I don't think there's anything wrong with using the G-Blocks. And really, from a workflow standpoint, we have found that it's easier to work with the shorter synthetic oligonucleotides than it is the G-Blocks, because G-Blocks, the yield upon order may not be high enough. If it's a quick screen, G-Blocks are great. But if we want to reproduce experiments, there may be a requirement for PCR amplification to get enough yield, and that was really the one of the limitations that we found there. It's still a great product for achieving this as a quick method to transfect the fragment in with the U6 promoter and see very efficient gene editing as well. So I guess it would depend on their workflow and their end application.
Yeah, there's a little bit of information on this on the website, which is www.idtdna.com/CRISPR. And if you go under the Performance tab, one of the things that's kind of interesting, there's a set of pie charts on there that talks about the kinds of editing that you see too, and it's a little different with G-blocks versus the Alt-R system, so that may be also some important information.
As you might imagine, it's you're transfecting in a DNA template, which could be after double-strand break occurs, if there's DNA template still hanging around, incorporated into that site of double-stranded break by a non-homologous end-joining pathway. And we definitely do see that when we use the G-blocks. And we do not in the case of the Alt-R CRISPR products.
Okay, here's one more question. This is a good question too, which is, how much is known about the immunogenicity of Cas9?
Of Cas9 itself. That is something interesting that we were, in fact, looking at, again, a piece of data this morning. We did not see any dramatic immune response from Cas9 only transfections, that is, transfecting the protein directly into the cells. Where we do see the immune response most dramatically is when looking at the in vitro transcribed sgRNAs, or the IVT products. Those are such that from different manufacturers and different sources and different sites, we see consistently upregulation of, I think we're looking at 4 different immunogenic markers, and see great upregulation of those particular genes by qPCR when cells are transfected with IVTs. We don't see any upregulation of those same markers when we transfect any synthetic oligonucleotides.
So we're basically out of time here. However, if you've asked a question and we haven't responded to it, we will email you, and if there's some answers that are particularly useful, we'll probably post those on the website as well as part of our FAQs. So yeah, I just want to thank everybody. We had a lot of great questions, a lot of great participation from the attendees. Garrett, that was a great presentation. Thank you for answering all the questions. And we will be making the recording available to you. We'll send you an email with links to the recording as well as to the slide deck. The slide deck is available right now if you want to go look at it. It's on our SlideShare site again. That's www.slideshare.net/IDTDNA. And we'll be emailing you shortly with the follow-up. So thank you, everyone. Thank you very much, Garrett.